Good evening, everyone. My name is Jane Pickering. I'm the Executive Director of the Harvard Museums of Science and Culture. Hopefully you can hear me, which includes the Harvard Museum of Natural History, which is the museum sponsoring tonight's event. And we have Pardis Sabeti, a geneticist here at Harvard, speaking with us, and I will come to that in a minute. But I just have to mention the usual housekeeping things. First of all, that you can pick up a brochure if you haven't already had it of the spring programs here at the Harvard Museums of Science and Culture. There are lots of different lectures and programs going on, and this is a great way to know about it. The other thing is that you can join our email list, and that's also another great way of finding out about all our programs. I'd also like to encourage you to think about joining the museums. There are wonderful member benefits for becoming a member of HMSC, free admission, all the usual things you might expect, and of course helping us to support the sorts of programs that we do at the museums, including tonight's lecture. However, speaking of support, this is my opportunity to thank Doctors Herman and Joan Suit in the audience, who very graciously sponsor our Evolution Matters series, which this lecture is the second, and there are two others coming later this semester. And again, there's a card with all the Evolution Matters lectures on it that is just on the table to my right. And finally, I would like to mention a lecture that is happening tomorrow, Intimate Inquiries Public Displays Situating Anatomy in Renaissance Italy. It's a little different for one of our lectures, and it coincides with a new exhibit at the Collection of Historical Scientific Instruments called Body of Knowledge, A History of Anatomy. And that opened last week, and this lecture by Katie Parks, a faculty member here at Harvard, is happening tomorrow at 6 o'clock. Not in our usual location, i.e. here, but in the Pfizer Lecture Theatre, which is in the Mallinckrodt Chemistry Building, which is just a little further along Oxford Street, just a little closer to Harvard Yard. Very easy to find, and that will be followed by a reception in the museum. So again, I'd like to welcome you to this. I also need to remind you that all our lectures are videotaped, and so you can see them. If you have such an amazing time tonight, which I'm sure you will, and you want to recommend it to friends who aren't here, then all our lectures are posted online at the HMNH website. I think I've said everything now I need to say. So I'm delighted to introduce you to Pardis Sabeti, who is Associate Professor at the Center for Systems Biology in the Department of Organismic and Evolutionary Biology here at Harvard, as well as the Department of Immunology and Infectious Disease at the Harvard School of Public Health. She is also a senior associate member of the Eli and Edith Broad Institute of Harvard and MIT. She completed her undergraduate at MIT and her PhD at Oxford as a Rhodes Scholar. She then went on to earn her medical degree from the Harvard Medical School as a Soros Fellow. As a computational geneticist, her expertise lies in studying genetic diversity, developing algorithms to detect genetic signatures of natural selection, hence our great interest in the Evolution Matters series, and carrying out genetic association studies. The lab which she leads focuses specifically on detecting and characterizing these signatures in humans and pathogens. She has been the recipient of numerous awards and grants, including a Burroughs Wellcome Fund Career Award, a Packard Foundation Fellowship in Science and Engineering, and a National Institutes of Health Innovator Award. Most recently, she won the 2014 Vilcek Prize for Creative Promise in Biomedical Science. She was also chosen as one of Apple's 30 for 30 years of innovation on the Mac, which I learned, for developing a tool on her Mac to determine if certain genes were the result of natural selection. And she took that Mac to Africa to work directly in the field, and this work led to new methods for curing and treating diseases like malaria and has improved thousands of lives. She has also been named a World Economic Forum Young Global Leader, a PopTech Science Fellow, and a National Geographic Emerging Explorer. The list goes on. I should mention she's a fan of the museums, which of course is the most important thing. And please join me in welcoming Pardis Sabeti.
Well, thank you very much for having me here tonight. I'm very excited to be here. As Jane mentioned, I actually have such a distinct pleasure. I get to walk through the museum about 4 or 5 times a day because I have a Fitbit and I'm walking a lot, so I'm doing a lot of walking meetings, and I'm connected through a set of corridors into the museum. So every meeting I now run into the museum, and at this point I've started almost to give tours because I'll start explaining things and then other people will follow, and so if sort of another career for me, I'd be excited to do it. So. But yes, so tonight I'll be talking about evolutionary forces in humans and pathogens. I'm going to just sort of set the stage a little bit, starting with, I teach a class to about a little over 400 undergraduates, and I found this is a teaching tool. We're going to try this. And so an engagement tool. I'm going to apologize if, I will be very careful not to pelt anybody. So just to kind of get you guys with the question, so can anyone tell me who those 2 individuals are? What's that? Oh, yeah. Okay. Well, you're gonna get the orange. Oh shit! I didn't warn you.
You're gonna throw something.
Yes. Yeah. That's right. I should have warned you that the orange is gonna come. But yes, and everyone deserves one. But I have to do it some way. So yeah. So Darwin and Wallace. Obviously, in this audience, I don't have to mention that. A lot of people forget Wallace, but I love him as does Andrew Barry. Yeah. And basically, in 1858, Darwin and Wallace first laid the paper that described natural selection. So even before them, well before them, lots of individuals were driving the theory of the ideas of evolution. But it was really when Darwin and Wallace laid out the principles of natural selection that started to really galvanize the discussion. And in 1871, when Darwin published The Descent of Man, that really sort of changed things even more so, because now we weren't talking about sort of plants and animals and other organisms around us evolving, but we as part of that whole evolution. But it wasn't actually until about 70 years after that that we had the first example of human adaptation. I'm trying to see if there's an orange opportunity here, but but maybe after that one I'll slow down. So in 1948, actually, J.V.S. Haldane, he's one of the founders of population genetics, and he, brilliant man, and he had lots of brilliant ideas, but one of them was very interesting is what they call the malaria hypothesis. And essentially, Haldane had looked around the world, and he had seen that there are a lot of red blood cell disorders, a lot of disorders like thalassemias, sickle cell anemia, ovulocytosis, and these all seem to be happening in regions of the world that were tropical. And in particular, regions where he knew malaria was endemic. And it made him question. We knew that malaria invades red blood cells, and he sort of wondered whether or not this was an adaptation. And just a few years later, A.C. Allison actually showed that that was true. A.C. Allison showed that individuals who carry the sickle cell protein, version of the protein, actually were protected from malaria. And soon after, just a few years after that, more and more of these traits, sickle cell, thalassemias, G6PD, ovulocytosis, began to be linked to malaria. So that was really the first elucidated example of human evolution. And since that time, and so here's the sort of a picture of the sickle cell, a sickled red blood cell that protects from malaria. And you know, one of the reasons why we know that selection is acting there is not just the fact that there's this hypothesis, but there's actually something happening in the genome that tells you an unusual footprint exists here. Something different was going on. And those kinds of patterns can be seen elsewhere in the genome, but there are actually very few examples that have been elucidated. So pigmentation, the lightening of skin as human populations moved out of Africa into different parts of the world with lower sunlight is believed to have evolved, causing individuals to sort of change their skin color. Also lactose tolerance, the ability to drink milk after we domesticated cattle, right? So all mammals drink milk, and for varying reasons, all mammals drink milk, and they take their mother's milk, but at a certain point they wean because there's no longer a source for milk. Or your mother doesn't want to give you milk after a certain point, right? So, but once we domesticated cattle, there was an external source for milk, and there was an advantage if you could keep metabolizing it over time. You had an extra source of nutrients. And so the way that we see these patterns, again, are not just this biological, very clear advantage that might exist, but also by looking into the genome. So the A's, C's, T's, and G's, the strings of letters that make up the human genome, within them, what we can actually see when evolution occurs, when a new mutation emerges and spreads through the population, is a footprint gets left behind. And I'll show you, it's actually quite a large footprint that gets left behind in the genome when a new variant spreads through the population. And in fact, actually, one of the challenges is to look within a large footprint and find the mutation that's actually driving what's going on. And we want to be able to set a magnifying glass and understand it. And the really interesting thing is that a lot of people, so I talk about the fact that I'm looking at human past, trying to find these things that are evolving. And most people seem to think that I must work with fossils. But we know the term is sort of been coined of fossil-free archaeology. And that's essentially what we do here. The genome provides this opportunity. We can take individuals today, so 100 individuals today. We can look in their genomes, and within those genomes is hidden an archaeological record that tells us something about our history in the past. And so we can take them and take the DNA from those individuals and look for mutations that are spreading through the population. So that's sort of the backdrop of everything that I do in my lab. But my lab is really spread into kind of 3 component pieces. We study human evolution. We're very interested in studying human populations in different settings. We look at the pathogens that are invading humans and affecting humans and understanding those, and we also develop methods and tools. And so I'll focus really on the first and the second, and then just mention some of the tools that we develop and the kind of resources you need to do this work. So again, let's go orient to a cartoon. So we have here, just I have a cartoon of individuals living in a generation. And at some point, there's a mutation that is going to emerge in one individual in their germline. That individual may pass that on, but pass any mutation that emerges in them on to their children. But if it's under natural selection, what it means is that individual carrying it is more likely to survive and reproduce. And if so, then they're more likely to pass it on to their children. And their children are more likely, who carry it, are more likely to survive, reproduce, pass it on to their children's children. So in a short period of time, it may spread through the population. And those are the patterns that we're looking for. Now on a genomic level, this is what the data might look like. Here's my chance for redemption in the orange throwing. Or we may go down lower. We'll see how it goes. So can somebody tell me in the audience, what are 2 major drivers of diversity in the genome? What are 2 things that are generating diversity in the genome? Oh, wait, yeah, is that from, okay, mutation, ready? Yes, all right. And what's another one? What's that? Sex. Yep, is that you? Okay. There you go. Yeah, it is sex. And what does sex allow? What happens when individuals mate in a sex? Yep.
Genetic recombination.
Yep. Okay. Oh, your computer. You're gonna. All right. Here we go. All right. So yep. So mutation and recombination. So mutation is generating new variants at different loci. So here's a picture of a chromosome, and there's positions across it. And here I'm just showing you a cartoon of 5 positions across a chromosome. And essentially, originally in a region of the genome, everyone's going to be the same. Let's say everybody is a green marker. And at some point, a mutation is going to create new variants in the population. So it'll generate these new purple versions of this thing. And recombination is this process by which variants nearby in the chromosome are going to get reshuffled with each other. So what ends up happening is you have mutation generating these new purple variants. And in the case of the selected one, I marked it red. And then recombination that's reshuffling those backgrounds. So most of the genome looks a little bit like this. It's a mosaic. But if a variant is so important and so valuable, advantageous, that it spreads to the population quickly, it'll happen so quickly, there's not that kind of time for the mutation and recombination in that local region to mix up what's going on. So you'll suddenly get a region of the genome where everyone looks identical for a long pattern. And that is sort of the footprint that we were talking about. And there's actually a few different ways of identifying it. So this is a little bit too clean because there will be some recombination happening at the edges here. But so this is something that moved very, very quickly. But so there's different patterns people use to find these things. One thing is called a long haplotype. And it's something called long-range correlations. What it means is you look at the variant that you're interested in, and you look to the left and you look to the right, and you kind of build this picture of what's going on. You see that the red variant's always next to a green variant, it's always next to a purple variant. So this thing is very conserved. It's got a very long, unbroken haplotype. But in this green variant, that's the common variant before selection, you can see that sometimes the green variant is attached to another green variant and sometimes to a purple variant. And then even if you break from there, you say, okay, green and purple, what happens next? You see there's another break point. So that's sort of a visual picture of what you're seeing is this breakdown. It's telling you something about how long the variant's been around. The more time it's been around, the more opportunities there are for these breaks in the background that exist on. So we're essentially dating the mutation. We're looking for things that have become very common very fast. Other types of patterns that we look for that are a little bit more possibly intuitive is just differences in populations, right? Human populations, okay, I'm going to actually pull it out again. How long ago did sort of human populations diverge from each other? Give it an estimate. You got to be from the back, don't you? Are we going to do this? You ready? Okay. all right. Ready? Oh. Sorry. We're going to, oh, sorry. Okay. Sorry about that. Yeah, but great. Yeah, about 50,000 years ago, and those estimates kind of move a little bit, but that's around that time frame. And that's great. Sometimes people are like millions of years ago, so it's great to check the background, but obviously this audience, again, is going to be very on point. And so about 50,000 years ago is not very long ago in geological time. So there haven't been that many differences within populations. And so in general, for most of the genome, we're actually very similar to other populations. But when you have these places where you have very stark differences, so in this is an example of a mutation that protects from Plasmodium vivax malaria. And you can see that the mutation has actually gone to about 100% in parts of Africa, but is absent outside of Africa. And that's another signal that you can see of natural selection. And a third one, okay, and let's see, well, we're one more try for an orange, and I think we're going to go orange-free for a little while, or clementine. All right, so is actually using relationships between other species. So one of the things that we're looking for, first I'll actually just ask the question, and then we'll come back, which is, how long ago did humans separate from chimps? 6 million. Is that you're another orange? I'm going to give it to you now, or are you going to? Here you go. All right. Excellent. Okay. All right. So about 6 million years ago, that's right. So that's a long time ago. And most of the variations that we see today in the human population actually happened sometime after that split. So one way we can answer the question, what's the ancestral variant? What was the variant that was there? Like, what was the nucleotide that was there originally in the genome? And what happened after is by looking at our close relatives and saying that whatever we find in them was likely the old variant that was existent, and whatever we find now is the new variant. And so in essence, we can look for these what we call derived variants. And in general, derived variants, in the absence of selection, are going to take a very long time to rise to high prevalence. So we're looking for things that are very common derived variants, or we also call it high-frequency derived variants. Okay, so that's just some background. You don't need to understand all the aspects of it to understand the rest of the talk, but we'll kind of come back around on this a little bit too. Okay. So those are the different types of techniques that a lot of people have been using to look for natural selection in the genome. And they've been developing those types of techniques for many, many years, but it was only since around 2000 where we really powerfully could look in the genome for these types of signals. And that's because of data like complete genomes of humans, chimps, and other species, large databases of genetic variants. We started finding all of those polymorphisms. Then we looked at large data sets of genotype data. That means that we took those polymorphisms and typed them in about 100 people or more, and got sort of the information about how they exist in the genome. And then now we're actually at the point where we're getting large databases of full sequence genomes. So this is really moving into that new era. And so all of these different landmarks have led us to being able to, for the first time, do genome-wide scans of natural selection. To look in the genome, and rather than have to make these observational hypotheses, very brilliant ones like the ones that Haldane make, we can actually have the genome tell us. So it's sort of the move from hypothesis-driven science to hypothesis-generating science. The interesting thing is, in this new era, we still actually have very few examples of human adaptation. So we were able to recapitulate things like the loci that we knew before, like lactose tolerance and pigmentation and malaria, but there aren't that many more candidates. One of the most interesting ones that came out a few years ago was looking at a population from Tibet, comparing them to neighboring populations in China, and showing that actually individuals from Tibet have a new adaptation, a variant that exists in them and not elsewhere, that is actually important for response to high altitudes. It actually changes the way that you deal with oxygen at a very high altitude. This is really exciting. But again, very few examples that have been known. And the reason why we have not suddenly figured out everything about the genome is the path to adaptation into like an elucidated adaptive evolution example is very long. It actually first involves developing a genome-wide test. These are each of the chromosomes along the genome where you develop tests and look for these peaks, these regions where something unusual is going on. But the thing about each of these regions is, remember we were talking before about how this region rose up in the population so quickly that there hasn't been a lot of time for that reshuffling. What that means is it takes the whole chunk of the genome up with it. And it's really hard to know who was the driver, what variant actually drove evolution, and who are the hitchhikers, all the variants that come along for the ride. And so that is the big challenge, is these regions are megabases long with thousands and thousands of variants, and you don't know which variant was actually the one that was important. And so we need to next move to localizing, to scanning these large regions and seeing where the signals are coming from. And then once we do that, it still may be that we're down to a smaller region, but we have to pinpoint the actual variant that is the driver. And we have to then figure out what is it doing. Because in this case, we don't know what we're even looking for. We're just looking for things that might be important. And that's the challenge. And so one of the things is I was actually writing a review paper several years ago, back in 2006, and I was actually trying to understand all the different tests people developed. And that sort of got me to understanding that they break out into these different types of signals, these long haplotypes, these derived alleles, and these differentiated variants. And while everybody, like in general in science, people have these sort of battles and they say, oh, your method sucks, this one's good, all of that stuff's going on, the more I studied them, the more I came from the long haplotype camp, so I was sort of heavy into that. But the more I started studying all of them, the more I realized they all actually have value. They're different signals that are emerging when a new variant rises in the population. So rather than select a favorite one, I thought, how could we combine all of these ones together? And two brilliant students in my lab, Sherry Grossman and Ilya Schlachter, worked with me to develop a test that uses all of these things simultaneously. So the interesting thing is that, all of these different signals will be in play, but the actual driver variant, a new variant that emerged recently in recent human evolution through the population, would have all of the signals, right? It's going to be on the long haplotype because it's the driver driving up that long haplotype, so it's got to be on there. And it's not always going to be that selection acts on derived alleles. It can also act on ancient alleles. But the ones we're going to detect by our methods are these recent variants that have spread as humans move to different populations. So it should be the derived allele. So we're looking for a high-frequency derived allele. And because we're thinking about things that are local adaptations to different environmental pressures or different infectious diseases in local environments, we're going to look for something that's also differentiated. So again, we're going to miss things that are more pervasive or older, but there's still a lot of things we don't know, so it'll get us somewhere. And so when you look, it may be that lots of things randomly get these high signals, but the selected variant itself should be something that has all of these signals. And so that's what we went after. And what we did actually is we developed a framework where we actually used the combined score by all of these things simultaneously to tell you which variant might be important. So let me walk you back to actually what our data used to look like. So these are actually the tests that everybody was fighting over. Mine's better, yours is better. They all looked a little bit like this. They all actually told you that something was peaking above the expectation of the genome. So most of the genome is more of a flat line, and then there's these places where these things are standing out with a higher signal. But it's really hard. It's sort of a very noisy signal within them to figure out what's going on. And when we combine all of these tests together into our simple, we call it a composite likelihood framework, it looked like this, which was very exciting. I was going into a faculty and thinking you're going to be wading into this. It was very exciting to get to this point, where suddenly the background really clears up. Because you might have a high signal by any one of these tests sort of by chance for being locally in the region, but the probability of having a high signal by all of these tests is very low unless you are the causal variant or you're right next to it. You're so close to it that you're like riding right along with it. And so here's an example of a region that holds a pigmentation gene, but it also has many other genes in the region. And the nice thing is it localizes to the pigmentation gene, and the high-scoring variant there is a phenylalanine to leucine amino acid change in that gene that is known to be associated with pigmentation differences. And we then, we did a lot of simulation work before this to make sure that our method was working on the theoretical expectations. But then we also applied it to a whole cast of regions that we had identified in our scans that had pigmentation genes. We thought that'd be a plausible candidate. But where the region itself was a megabase, so that means 1 million nucleotides long, and it wasn't clear that the signal would actually go to the pigmentation gene. But in every case, it did. So here's a bunch of different genes, and it's always falling right on top of the gene that we're interested in. The only one that's a little bit different is this one, which is kind of interesting, because it's a very well-known pigmentation gene, and the top signal is right on top of the pigmentation gene. But there's something going on here, and this is actually the main transcription factor binding site for this gene. So it looks like there might have actually been 2 events. So one event that might have arisen and another one that came after. And so once we kind of identified that this was working really well, the exciting next step was to apply it to the rest of the genome and see what we'd find. And so we love these fadeaway shots. We always show the first, the before, the after. It's sort of fun that these are a lot of different regions. There's just like example of different regions in the genome that we are scanning through, and these are our old tests, and this is what I had thought I was going to have to mine through. And as we developed the tests, suddenly they all look like this. And there's still a number of candidates that are in the region, but at least we're getting to the gene or the locus, right? And it's much easier to start at least hypothesizing about what's going on at the locus and then really trying to dig in. So here's an example of one of the ones we went after. So again, this is the original signal. It's 800 kilobases, so 800,000 nucleotides across the genome, and about many hundreds, thousands of variants within that region you're trying to mine. But when we run our test, it gets a lot simpler. And this variant right at the top, the very top signal, is a valine to alanine change in a gene called EDAR. And actually, so EDAR, Alex Shear here was the one who got me very excited because he knew EDAR very well when he saw this in my list and said that actually David Kingsley's group out at Stanford had already found something really interesting about it. So this is a picture of stickleback fish. Stickleback fish are really interesting from an evolutionary perspective because they originally were in seawater. And over again, they kept moving out of seawater into different freshwater ponds and populating them. And so there's a very good example of sort of this, a lot of independent events that we could look at as to what happens when fish move from seawater to freshwater. And what they always kept doing was they kept losing their scales because they didn't need them perhaps, they were expensive to make, and also maybe this way you could absorb nutrients better, there could be other advantages. But this was a clear thing that was going on, and he localized it to be repeated mutations in the EDAR pathway, so the EDA pathway. And this is ectodysplasin. It's basically what's expressed on your surface. And in fish, it regulates scale, but in humans, it regulates sweat, hair, and other ectodermal things, also teeth, the formation of teeth. It has a lot of different effects, actually. Here's actually, soon after we published the paper, a couple of really exciting papers came out associating the variant that we had called the valine to alanine change with different phenotypes. The first one is actually hair thickness. So this is actually, if you look at your hair and you chop it in half and look at it under a microscope, the thickness of the actual hair. This is the original, the ancestral way that it looks like. If you have 2 copies of the ancestral, it looks like this. This is the size of your hair shaft. Well, obviously blown out, but the relative size of your hair shaft. This is if you are a heterozygous, if you have 1 copy of each. And here's if you're homozygous with the derived allele. It's about twice the size of the original. And if you look, actually, one of my friends Asian friends had basically said that he was looking under hair, and his hair under a microscope looks like a fiber optic cable, and mine is like this, like it's invisible, so fine. But essentially it is that many Asians have very, very thick hair on their head. And also, as a classic thing in anthropology was something called the oriental cusp, which has now been moved to the more appropriate sort of Asian tooth shoveling. And this is essentially that in the back of your front teeth, if you kind of look at the ridge behind your front teeth, for most people it's smooth, but for many Asian individuals, they have like a ridge in the back. And that actually is also driven by this mutation, interestingly enough. So we started to see that there are a lot of different phenotypes that are being, so are you testing, yeah, all right. Yeah, people are, like a lot of my, when we do this, like people start going to the Asians and trying to feel behind their teeth. So don't go after her. But yeah, so there's this interesting pattern that you see. And we wanted to see whether or not this was just an association. Just means that if you have it, you're more likely to have this. You really want to test to see, does this single mutation itself make this big change? So for that, we need mouse models. And I worked with Cliff Tabin and Dan Lieberman here and Bruce Morgan as well. We sort of developed this great faculty collaboration to go after this. So we took the EDAR mutation with the original valine, and mice have very similar EDAR genes, so you can do this. It's very conserved. And we placed a single alanine change into a mouse model. And we looked to see what the differences are that exist. So just in a cartoon fashion, I'll just show you that what we did find is actually that the hair got thicker. So that single mutation, place it into a mouse, and you can actually make the hair on the mouse all over its body get thicker. It also, interestingly, which is not good for the reporting of the paper, but for the popular reporting of the paper, but is a true thing, it actually changed the shape of the mammary glands. It made the mammary glands a little bit smaller and the branching a little greater. So this is a very pleiotropic gene, meaning it affects lots of different things. And then the other one is that it changed the eccrine sweat glands. It increased the number of eccrine sweat glands in the mouse. We can only look at them in the foot pads of the mouse, but essentially the foot pads of the mouse got more sweat glands. And then another thing that we did actually was just try to find out, okay, this mutation seems to actually be driving all these changes. Where did this mutation come from? How did it happen? And we can use genetic analysis to show that the mutation appears to have emerged about 30,000 years ago. And it's sort of the high probability that it came out of somewhere in central China and then spread throughout different parts of Asia and into Native American populations. And you can see a lot of Native American populations that they may look different, but they have a lot of the same phenotypes of hair and sweat. And so we see this pattern emerging. So one of the things we wanted to do after that was then say, okay, let's go back. We already knew about the hair and the teeth differences in humans, but now my postdoc, Xizhe Wang, had done his original work in China in Shanghai, and we collaborated with Li Jin at Fudan University to look at a cohort that came somewhere around in sort of that central China area, but here on the edge, the Taizhou Han cohort. And what we did there is we developed a study where we looked at lots of different patterns of hair and sweat. We really wanted to focus in on this sweat thing because this was a new trait that hadn't been studied before. And what we found actually was, now most of the Chinese individuals that we were studying didn't have the ancestral variant, and very little of the ancestral variant. It's almost at 100%. So you don't see many individuals that have two copies of the ancestral variant. So that makes it a little more challenging. So we can only really compare the heterozygote with the homozygous individuals that carry two copies of the selected trait versus one copy. But you can see here that actually the number of active sweat glands is a lot higher. And it's actually, I always say, it's got such a beautiful pattern. Like we're sort of sweating in blotches here, and it's almost as if you have like many, many more sweat glands that are each releasing a little bit of sweat from them. So this is another type of phenotype that we found. So a lot of times when I talk about this work, this is not actually, I am an MD, and I really am interested in infectious disease. I didn't end up wanting to become a hair specialist. But people ask me, how is this possible? Why would this be under evolutionary pressure? And so I kind of go back to this slide. And I say, if you were to ask, and let's pull out an orange for this, but okay. If you were to ask, what are the differences? So what's different about these families? What do you think of the first thing like a kid would say? Who wants an orange, actually? This is everyone might have an answer. So anybody hungry? Okay. Nobody wants to take a risk. Oh, come on, somebody in the back. I'm going to just run down there because I got in trouble. So here we go. Oh, there we go. Sorry, it's still boxed out. It's rolling. It's coming down. Somebody's going to get an orange. I'm going to get two more and give it to those guys because I think yours rolled off as well, and you deserve one. 1 second. So yeah, if you ask, and I'll just keep talking as I hand out these oranges. If you ask, what's different about these families, most people will probably first come out and say hair, right? That's sort of dramatic. Then you have some shorts, you have some glasses, you get some different posturing. But in general, that's a very major difference in how we've changed over time, right? And what is interesting about that is humans have changed a lot. I often say that you think all the other animals look at us and think we look so weird. I mean, we don't have any skin on our hair on our bodies, and we have these long hair. We look very bizarre, but we've really changed the pattern of our hair. And what's interesting is that we're the only mammals that actually use eccrine glands. These sort of have these deep eccrine glands all over our body in order to thermoregulate. And one of the things is it's likely been very important. Any animal many animals can outrun us at a short distance, right? Cheetahs can outrun us at a short distance, but we can outrun any animal at a long distance, right? They can't do those marathons that we do, and that's because most of them would die at some point of a heat stroke, because they're only sweating from their palms and their toes, and they're using panting. But we've really changed that whole pattern. So thermoregulation is incredibly important. You'd imagine it's very important, particularly during pregnancy and things like that. So changing to your environment will be a very big driver of human evolution. All right, so now let's get to the infectious disease, which is more where I feel comfortable. So I'll start there. So here's another one, a leucine to phenylalanine change in the gene toll-like receptor 5. Toll-like receptor 5 is one of the things that's very important in the innate immune system. It's sort of one of the first line attacks. And here's this amino acid change that's high scoring in our region that's right here in the main binding pocket. So this is sort of a dimerization of these two subunits of toll-like receptor 5. And this mutation is sitting right in the middle of that. And interestingly, another thing you can do to start saying, is this mutation important, is just look, is it conserved across lots of mammals. So this is looking at humans as well as Neanderthals, but then also a lot of different mammals, then down to other kind of vertebrates like chicken and frog. And the interesting thing is that leucine mutation is highly conserved. So it's all across mammals. All mammals have that exact leucine mutation. It hasn't moved away from that. And then the interesting thing is that in other places, the teneric and the platypus have mutated away from that, but they also mutated to phenylalanine. So that's sort of like locked into that position, tells you that could be convergent evolution or could tell you something about what's going on there. So we found it interesting. And so just in a cartoon fashion, I'll show you what we did. What we can do, the same way that we placed the one mutation in a mouse, you can actually place a single mutation in just a cell culture system, where you can do it in sort of not a native environment, but within the sort of the protein structures. We can actually take it and place that single mutation, so into the protein itself, and express it in cells. And then the interesting thing about toll-like receptor 5 is it's sitting here on this. It's a receptor that's sitting here on the surface, and it's actually the main line of attack for flagellated bacteria. So these bacteria have these flagellae here, and in it there's a protein called flagellin. And this is the only actual protein that recognizes these flagellae. And what happens is that flagellae, the flagellin, the protein in it, binds to TLR5 and then drives a signaling cascade that's really important in our immune response. And as it turns out, when you have flagellin binding, depending on which version you have, you get very different signaling. This one gives you a lot of signaling downstream, and this one gives you actually diminished. So the thing that was actually advantaged, appears to be advantaged, was actually diminishing your response. And as it turns out, if you knock out this gene altogether in mice, they're protected from Salmonella typhonurium, which is very related to typhoid fever in humans. So actually diminishing your response may be protective from having a disease from these flagellated bacteria. So these are the kinds of things we're finding. And I have to say that, the process by which we go for is we start with these genome-wide scans, then we try to find map the variants, then we use something called computational annotation, meaning that we one by one look at every variant and try to figure out which ones might be valuable, and then we run these experiments and then try to get to adaptive hypothesis. This part of it is what takes a long, long time now. So this is now where the slow part is, and so we have to be very cautious. We're sort of 2 for 2 for what we found, but we're careful. The mouse takes 3 years to make, so you don't want to be wrong about that. And we're moving right now to making high throughput functional experiments to do that much faster. And so these are the pictures. Just we've added a couple things, but it's still pretty sparse. And there's all these other things in there that we're trying to figure out what they do. One story I'm not going to have a chance to tell you about, but it's a very cool story, is that we did the same kind of analysis in looking at cholera. Does anyone dare? Does anyone know what that picture is of? Yeah, it's a delta. It's a delta. You are just an orange. Ready? You want to hand this out to some people? But yeah, this is a delta. This is the Ganges River Delta in Bangladesh. It's actually the epicenter of Vibrio cholera and has been for thousands of years. And so we know that cholera has been infecting the population in Bangladesh for thousands of years. We know that cholera is highly fatal, very prevalent in those populations. All of the kind of things that are important for natural selection to act is something that's prevalent, something that's fatal, something that's been around for thousands of years, and something that has a genetic component. It had all of these things, so we actually pursued looking at signals of natural selection in cholera, and we found some really exciting stuff. We have some new stuff coming out that sort of looks great. So, in the matter of time, I'm just kind of giving you some snapshots. But essentially, where we're at is that we started by really trying to develop methods to find all of these regions, but that's not really getting us too far. But now as we develop these new tests, we can start saying, oh, they're actually localizing to specific genes. Some of them are localizing to what we call EQTLs, which are things that change the expression of those genes. We also have things that are localizing to regions that don't have any genes at all. But the more and more we look, the more we realize those things are actually themselves important. And things that change other aspects of the gene, like something like called splice site mutation, so how the gene is transcribed and translated. And we're also seeing that things are congregating into different pathways, like metabolism and infectious disease. Unfortunately, we're seeing some things that are going to get thorny, but we'll sort of explore and look at it very carefully. And then other things that cause pigmentation, sweat, and hair is one of the major drivers. So now to pathogens, which is, I'm going to give you one story of pathogens and then just tell you about the methods and tools I developed. So the actual strongest signal, we're looking at a population in Nigeria, and the strongest signal that we found was actually in a gene on chromosome 22 called LARGE. And our CMS signal, our composite of multiple signals signal, points us to one section of this gene called LARGE. So I didn't know what it does, so I went looking. And it's very interesting. Mike Oldstone out at Scripps did some very interesting work where he showed that LARGE, this is the gene LARGE, and it's named so because it's a very large gene. It's 700 kilobases long, but it also does something interesting. This is alpha-dystroglycan, which is important in sort of muscle development, but it's also expressed all over the body. And LARGE actually binds to alpha-dystroglycan. And when it does, it causes these little glycosyl groups to be added to it. And then those glycosyl groups signal for the protein to be cleaved, and then that protein cleavage signals for the protein to be moved to the cell surface, where it actually is a receptor for a number of things. But interestingly, a lot of microbes then co-opt it to enter into cells. And there are a lot of different microbes that co-opt it, but the ones that Mike Oldstone had shown, was actually arenaviruses, like this virus called Lassa virus. And essentially, Lassa virus uses this now as a receptor to get into cells. If you knock out LARGE, you stop this whole process happening, and LARGE cannot enter cells. So it was interesting because suddenly I had gone to medical school and I had never even heard of Lassa virus. I went back and it was like a footnote in one of my tables. And so how is it that possibly this virus could be one of the major drivers of human evolution, the single strongest thing that we found in this African population? So the next slide is going to tell you a little bit about Lassa. There's an image that's going to show up a couple times in my talk. It's a little disturbing. I just want to warn you. Look away if you want, and I'll say again when it comes off. So here's the next slide. This is what Lassa looks like. It's a hemorrhagic fever virus like Ebola, like Mopea, like Marburg, a deadly virus that causes this overt bleeding from lots of different, can cause this multi-system organ failure and this large-scale bleeding. So this is one of our patients. Now it's going to come off. So this is a very deadly disease. It's interesting because it's actually pretty widespread. So we call this a biosafety level 4 because it can kill you within 2 weeks and it can be aerosolized, can go transmit in air. So you have to work in it in biosafety level 4, meaning in this very contained research settings. But the interesting thing is it's actually very widespread. So unlike a lot of other biosafety level 4 things that you think are only happening very, very rarely, it's actually known to affect many different populations. In Nigeria, 20% of people tested had antibody evidence of exposure, having been previously exposed. That's millions of people. In Sierra Leone, Liberia, and Guinea, in some of the populations that have been looked at, 50% of the individuals have antibody exposure. Now, there's some sensitivity issues with those or specificity, but there's a lot of evidence that this thing has been pretty widespread. And interestingly, it makes sense that it's widespread because that cute little mouse who might come out in your kitchen, comes out in kitchens all over West Africa, but is actually harboring Lassa virus. So the Mastomys natalensis has co-evolved to essentially breed Lassa in its blood year-round. In some of these populations, 40% of the rodents are actually carrying Lassa virus. And so what we ask is actually, you have something that's so widespread that is being carried by this rodent that's moving all around this area, could there be genetic resistance if it's been around for a while? And so that's essentially what we went to look for. And we worked with individuals from Tulane, actually have been working for some time in Sierra Leone. We actually partnered with Christian Happy in Nigeria and moved to 2 hospitals in those 2 countries where we began to explore. And essentially, here's the Lassa ward in Sierra Leone, and here's one of the hoods that we do our work in. These are the diagnostic facilities that we use to study it. This is a pregnant woman. So pregnant women, the fatality is about 100% for them and their baby. But once we start doing diagnostics, many of them start to survive. Because there is a drug, ribavirin, that decreases fatality in a published study from 55% to 5%, only if captured in the first 6 days after sort of the symptoms begin. But at that point, it's very hard to detect what's going on, because the original symptoms are sort of fever and malaise. And so, in the process of doing the research, we had the ability to make an impact by creating diagnostics on site that would be able to work with these patients. Now I'm just going to go back again. Oh, actually, one slide before that. So essentially what we did is we're working in these 2 sites. We're also sequencing from the reservoir, from Mastomys natalensis, from the human patients, and we're sequencing those individuals. And the kinds of things that we found is actually we've sequenced the virus, this small sort of viral genome that has sort of 2 chromosomes or 2 segments. And what it looks like when we sequence viruses from Nigeria and Sierra Leone, and also there's some published sequences, 1 or 2 from the middle of those 2 regions, is that the virus is at least, and this is a minimum date, at least over 1,000 years old, around 1,185 years ago is a minimum date of the virus in Nigeria. And it actually seems to have spread into Sierra Leone only about 150 years ago. But it's been circulating in Nigeria for a very long time, which is sort of links with our idea that it's something that's genetic resistance is evolving to. And also, this kind of work, when we're sequencing the virus, also allows us to build better diagnostics, and our collaborators at Tulane are doing that as well. So what we're seeing here is that this virus that I hadn't even heard about in medical school actually is a virus that seems to infect many, many people. And we're seeing at the sites that we work in, we have many, many patients coming in, hundreds of patients coming in each year. And it also seems to be very old. So now we're dealing with something where evolution could be acting. And we may just start to ask a question. I'm going back to the slide again. So here's another warning of this slide. This is essentially what we're looking for when we think of a biosafety level 4 emerging virus, right? This is sort of when you think when the public starts to panic, they think that they're going to see this. And this is one of our patients, and we do see patients like this. And now I'm moving off the slide. But most of our patients actually look like this. They come in with maybe, they just have malaise. They feel unwell. They may have a little bit of bleeding here and here, may not. They may have fever. They may not, even though it's called Lassa fever. But this also has the same fatality rates. In Sierra Leone, where this patient comes from, fatality rates of patients even looking like this is 80% without treatment, is what we're seeing. And so the interesting thing we started to ask ourselves is, what are we actually dealing with here? Are we dealing with emerging disease or emerging diagnosis of diseases that are actually already circulating widely in clinics and has been doing so for many, many years? So this image, developed by Stephen Geyer in my lab, is one that is he is actually originally was a picture of the Congo River near where Ebola was first discovered. And Stephen, as an artist, designed it so that he actually drew the Congo River into the shape of the Ebola virus, to kind of pose the question that if we look from a different angle, if we look more carefully, are all these viruses that we worry about that may one day come, are they actually already here? And have they been here for a very long time? And so this is the kind of place where this evolutionary work can take you. And so one of the things that we like to say is what's really exciting about this is we started at the Broad Institute, is the genome center where we did the sequencing. And we were doing this sort of sequencing here, but it moved us, and our collaborators here at Harvard, to then build diagnostics that we took to the field to start trying to collect patients and start doing research. But when we actually allowed for local diagnostics, what we did is actually enable the community. We enabled the doctors, and we created resources. The community became engaged, the doctors became empowered, and we keep getting more and more patient samples. And not just from Lassa fever patients, from other patients. People were so excited that they could be diagnosed for something that they kept coming to us from hundreds and hundreds. I mean, I actually, let me hold that. I mean, they're diagnosed for a number of things, but this virus in particular, they knew they couldn't be. So there's certain things, so something was not the right word, for this thing that was deadly. And so it made them incredibly excited and empowered, and more people came from hundreds and hundreds of kilometers away. And we started to essentially become the sentinel site for what other things are infecting the populations. And so this has led us to my collaborator, Christian Happy in Nigeria, has just won a very large World Bank grant and an award from the NIH to actually build more capacity to do this microbial surveillance in Africa in partnership with us. And then that just brings us to me to mention the different methods and tools. All of this work is really developed. At the core, we're a computational lab. So we develop technologies to mine large-scale data sets, to mine the human genome, looking for these long haplotypes, to look for patterns, looking for sort of continental spreads, and also to do collections in even remote settings. And that's sort of the core of what my lab does, is develop these types of methods and tools. And we also look at an array of microbes. So I talked to you a lot about Lassa, but we actually work very extensively on malaria, on seeing how malaria has evolved and changed over time, as well as Ebola and cholera. And with that, this is just the scan. So these are the individuals to acknowledge on the two papers I talked about more, which is the EDAR variant and the TLR5 variant. Sherry Grossman and Ilya originally made the CMS method and teamed with Christian and Shervin here to really work out what was going on there. And Jana and Sija really drove the work showing that EDAR works. And these are major collaborations. The wonderful thing about being here at Harvard is just the extraordinary collaborations from so many different areas. Cliff Tabin, Bruce Morgan, Dan Lieberman, who's presented here before, and collaborators in China and in Europe as well. And of course, the mouse, one of the most important individuals in the study. Also individuals, Eric Lander's been my advisor for 20 years, so that dates me. But the many collaborators that worked with us on the toll-like receptor work. Eleanor is the one that's driving the cholera work here. Steve Schaffner is a longtime collaborator. The work, sorry, I always do, it's almost like the Academy Awards where I'm like, I gotta, kick me off, but it's just, science is not done by individuals, it's really done by teams and partnerships. So Stephen is the wonderful person who led the emerging virus paper and also made that beautiful image. Christian leads a lot of the Lassa work and a lot of the studies there, and also collaborated along with Matt on a lot of the emerging virus work and the Lassa work. And then I can't name everybody in my lab. I'm not going to hold you here, but I always just sort of show this was my lab in 2008 when I started it. This is Dave, who developed a lot of our algorithms, who missed the photo and got an American flag and a ridiculous treatment. The next year we got a little more artsy about it, kind of had a Gossip Girl theme. Sija missed the photo, and it was made into a portrait. Jana's creeping out from the bookshelf. The next year, just to show you, we got, these guys are so creative and everything. This is just to let you know that this is actually done, actually underwater, although we're not under the ocean, but we did, so the photoshopping did occur, but not all the way. Next year, the Academy of Athens, always a good favorite. So we reenacted the Academy of Athens. People who missed the shot became decapitated heads. And we had a good time. And we sort of Gangnam Style the next year. Kate and her horse were obviously photoshopped in, although the horse is Gangnaming right there. And Saturday Night Live for the next year. So we're sort of reenacting Saturday Night Live. This is what's also happening in the card. So it's getting more and more intricate on the inside, as if there's not enough time. We actually got some celebrity appearances. So Chris Martin, this is a Coldplay playing on the show. Chris Martin actually is holding our shot there. Seth Meyers from Saturday Night Live is holding our lab T-shirt. So they actually took pictures for us for the shot. And Sarah Palin, the whole crew. So all your favorites from SNL. Then the Nigerian collaborators. So this is our institute in Nigeria. Christian Happy is my long-term collaborator. Extraordinarily beautiful people. Actually, many of them will be here this summer for a training program that we have. And these are the collaborators in Sierra Leone, also really photogenic, right? But amazing, beautiful people, but also really great spirits. Eric and the whole team and the broad that I work with, the Lassa fever community, which fits in a room. This may be half the community, but still fits in a room. And then I always thank the many funders, the individuals, and I will end with a picture of this and a song that I actually recorded with my African collaborators, because they're not just beautiful, but this woman here makes me cry every time she sings. They all have beautiful voices. So when they were here last time, I was like, I just took them into a studio and recorded one of my band songs with them. So this is the girls who are singing, and this. Oh no, music went away. Did the music go away? Let's see. Yeah.
Come like waves.
other Lassa patients and for each other, and thank you so much. Thank you. Second down or so? Oh, Sue.
I'm sure everyone has lots of questions from that very moving presentation.
Thank you. Yep.
Can you say a few words about your Ebola work?
About the Ebola work, sure. So, well, we have just a starting operation in Ebola, but we working with USAMRIID, the U.S. Army's Research Institute for Infectious Disease, and we're studying how the virus evolves within the course of infection. We've sequenced patients from clinical samples from Gabon. We're also looking at a study that they did of non-human primates that were infected with it, understanding how the host responds to it and how the virus is evolving over the course of infection.
Thank you very much.
Yep. Yep.
You do your work, or any thoughts on applying your techniques, developing techniques for epigenetic studies?
For what kind of studies?
Epigenetic studies.
Epigenetic studies. We use epigenetic data in sort of understanding. We use the kind of data coming from the ENCODE project and the atlas of all these different elements to see if any of these effects might be driving other kinds of things, but we ourselves have not focused in that. There's a huge effort happening at Harvard and at the Broad driving epigenetic studies, but we're kind of more watchers of it, and at some point we may engage, because epigenetic studies are studies of all these different elements, the non-coding region of the genome. So basically we're saying only 2% of the human genome actually codes for proteins, and there's all this other stuff going on. And not only that, there's all these things that are happening that are changing over time. So Lamarck actually is being shown to actually be right about some things, that some things actually do change over generations just by our activity. And so there's a really fascinating thing of understanding this whole landscape of the genome that we know very little about. Yeah? Was it you that had a question? Yeah.
Okay. I did. I was going to ask you about a virus that they just defrosted, 30,000 years old. Anything about that? They might be related to the Ebola virus?
Yeah, it may well be. I mean, so I know about it, but I don't know any details about it actually at this point. But it's a very exciting time. There's a lot of work that's showing actually how ancient these viruses are, and that they've been around for a very, very long time. Ebola virus also has been around for at least sort of 10,000 years. And interestingly, their precursors have been around for millions of years. Ebola virus is actually Ebola-like virus has actually integrated into mouse and rat genomes before they split. So it sort of tells you that millions and millions of years ago, these viruses were infecting individuals, and some of them even integrate into the genomes. So it's likely that Ebola and Ebola-like viruses have been around for millions of years, and that Ebola itself has been around for thousands, many thousands. Yep.
Are there any signatures of resistance to Lassa fever among the many people who never developed the disease but who have antibodies?
That's what we're in the middle of studying right now. You can imagine that it's been very challenging to set up a lot of these studies because we're trying to do work that you have to do in a biosafety level 4 cabinet, but in a very rural setting. So we have now collected a few hundred individuals, and we're analyzing their genomes. And we actually have preliminary evidence that there is. Yeah. And so then, yeah.
Could some of your techniques be applied maybe to gut flora that have evolved and co-evolved with humans?
Can they be applied to
Gut flora in humans?
Oh, gut flora. Aspects of the tests that we develop can be used for sort of epigenetics or for microbiome work and all of that, but they're different. Each one requires slight changes, but some of the principles, like looking for things that are evolving and changing, will exist. So we're really specializing ourselves because you want to sort of develop something that's best for the type of selection you're looking for, but a lot of the same kinds of methods can be used for sure. Yep.
Could you comment on the relationship between computational and empirical methods? I'm a computer scientist. I'm concerned about the growth of big data.
Okay.
And people trying to determine causality from models, which you cannot do.
Right.
So the question is, you're using a lot of computational models. Do you use it like a flashlight to then induce empirical studies?
Yeah, no, it's a very good comment and a very important concern because, like you said, when you start dealing with big data, it's exciting because you feel like you can make all these discoveries, but one error in your algorithm and you are finding nonsense. In your model, absolutely. So we actually, this is a joke that maybe only you would like, but I remember reading a lot of, I was reading a lot of Onion articles while like doing many simulations, 16 hours a day on this, and with my friend who I was sitting with for 16 hours every day, I at one point turned to him and I said, I have a new Onion title, which was Finding Natural Selection in Neutral Simulations. So, but basically, like, I was starting to find selection even in the models of no selection, and so you can get yourself in trouble, but yeah, so.
I mean something even deeper than having an error in your model.
Yeah.
Other people believing that the results of computation, it points to causality.
Absolutely, absolutely.
And that's not possible.
Yep, and so to answer that question, we use it as a flashlight. And that's why we're doing all these functional tests that may take years, because we're just moving one by one to prove each thing's out. And even once you prove it out, you have to still understand what it's doing and how it could be under selection. Should we maybe take like one more? I don't know. I'll stay here as long as people want, but I don't want to make everybody sit. Yeah.
I would like to ask if you could comment on the evolutionary aspect of how is it that this disease is 100% fatal, 80% fatal, and all the people who are susceptible are already out of the seed.
Mm-hmm. So how is this thing so fatal and all the people are not out of the seed? It's a good question. I mean, so very few people are likely actually susceptible in those populations, because if you look at the exposure rates in Africa, in Nigeria, you're actually seeing millions of people that have likely been infected. So actually the people that are coming into the hospital are probably the outliers in the population that randomly have one of these mutations. It actually takes a really long time for any mutation that's under the kind of selection we're talking about to get to 100%, because often it's sort of enough to get it down to a certain level that it kind of rides up pretty quickly, and then it sort of says, okay, that's enough. It's now more sporadic, and so those genes will kind of keep passing down. So what you often see, even with lactose tolerance, it's clear it's better to have more ability to drink milk, but it actually went to 80% and is now sitting there. So you do see that often, that maybe in hundreds of thousands of years you'd get to 100%, but a lot of times you just say, okay, that's close enough. We're moving. One of your children will probably get through. You might not. So there's like a few copies. Not good for you, but as evolution's not thinking about. It's more like in the gross, if your genes are being passed on, some of those slip through. So we do think that the vast majority of people, particularly in Nigeria, are resistant to the virus. But there are still some individuals. But I think that's the more rare, and it tells you actually how much exposure is going on. But yeah. Yeah, yours, yeah.
Were there differences in exposure rates between Sierra Leone and Nigeria that reflected the perspective and the side difference between the two? You said there was about 1,000 years.
Yep. So the exposure rates we don't really have a good sense of, because the exposure rates are dependent on how we're actually capturing information, and right now it's too patchy to know. But we do see a lot of very interesting differences between the populations. Our fatality rates in Sierra Leone, even with treatment, are extraordinarily high. They're still sitting around 70%. And in Nigeria, with treatment, is 33%. So there's something going on there that's still problematic, because it should be lower than that with treatment. But people are coming in more and more in later stages. So we're seeing it seems to be more fatal in Sierra Leone, which may associate with the fact that there might be more genetic resistance in Nigeria. But we're also studying the virus itself, and the virus has mutated a lot since it left Nigeria and went into Sierra Leone. It seems to be opening a new niche and changing. So we don't know exactly what's going on, but something interesting is going on. So I can stay around, and maybe I'll. Okay, sure, yeah. We don't know at this point. So the question is, is the virus, we know that the humans may be evolving to the virus, is the virus evolving to the humans or to that specific mutation? We don't know about that exact mutation, what's going on. In malaria, we see instances of that, like a receptor that seems to be kind of the point of some activity. Here, we don't know exactly what's going on, but we do know that the virus has evolved a great deal as it's moved into Sierra Leone, and we know that the humans are evolving as well. So that's sort of, it's this process of kind of unraveling what's going on. It'll take a little while, but there's going to be a lot of those instances of the receptor changing and then the virus changing and then back and forth. Thank you so much for your time. I really appreciate it. And thank you. Thank you, Jane.